Good morning, my dear students. So, myself, Dr. Shibu. What we are discussing is it is about application of recombinant DNA technology. In the last class, what we discussed is definition of recombinant DNA technology and overview of recombinant DNA technology, and we started with certain aspect of our DNA technology and the product. Okay, such as metabolic engineering and about protein, native protein improvement. These are all the things we discussed in the previous class. Now it is an application about recombinant DNA technology. So and the major point which is involved in the recombinant DNA technology, it has been widely used for the improvement of native microbial protein. Okay, so the improvement of native microbial protein or the product, which means the product or the protein which is produced by the microbial system itself. So in order to improve that, for example, if a microorganism is producing an amylase, we need to improve the quality and the quantity of the amylase. That is one criteria of recombinant DNA technology. Or if it is producing any antibiotics such as penicillin, chloramphenicol, streptomycin, whatever it may be. Okay, so it may be improved with the help of this recombinant DNA technology. Then frequently, this have been involved in self-cloning work where chromosomal gene is inserted into a plasmid and the plasmid incorporated into the original strain and maintained at high copy number. Then the next point what it is mentioned about the application is majorly it is involved in self-cloning work where a chromosomal gene, the gene of interest, is inserted into the plasmid. Then gene of interest is inserted into the plasmid, and the plasmid is incorporated into the strain, original strain where we are going to grow it, and we can maintain high copy number. That is one aspect. And the first application of gene amplification to the industrial strain was improved for the enzyme production. Okay, so the first application of this recombinant DNA technology as well as the gene amplification in an industrial level was to improve the enzyme production. Okay, the production of beta galactosidase, penicillinase, chloramphenicol transacetylase, and aspartate transcarbamylase. Okay, were all increased by transferring the plasmid containing the structural gene into the recipient culture, especially when the plasmid replicated faster than the host chromosome compared with that of normal host chromosome. The plasmid which was transferred or a new plasmid which is having a gene of interest, that will be replicated more fast and the production also improved. We can get a high copy number. High copy number of the plasmid leads to high production of the product. Okay, so the development of the first commercial recombinant lipase for the use of washing powders was achieved by Novo Nordisk. Okay, so the development of lipase have been done with the help of recombinant DNA technology. And the first successful application of genetic engineering technique to the production of amino acid was obtained in threonine production with the help of E. coli. Okay, so Dept in 1982 investigated the production of threonine by a threonine analog resistant mutant of E. coli strain K12. Okay, so the first successful application of genetic engineering and the production of amino acid for the production of threonine has been done. Okay, so these are all few application about this recombinant DNA technology, and it is mentioned the application of recombinant DNA technology to the development of tryptophan producing strain has been particularly valuable, and relatively low productivity was achieved using the classical mutation or selection approach despite attempt over a 40 years period. Okay, so for the over 40 years of period we are following the classical mutation for the improvement or for the production of tryptophan but by using the recombinant DNA technology we can develop the tryptophan production that we can get a more success compared with that of classical mutation. Okay, so that's all for today. So hope you will understand about application of recombinant DNA technology in strain improvement. So if you have any queries or any doubt, you can contact me. So thank you all. See you in the next class.